Hello class, today we are going to learn about the innate immunity, non-specific defense of the host. Immunity, also called resistance, is the ability to ward off disease caused by microbes or their products, and to protect against environment agents, such as pollen, chemicals, and animal dander. Innate immunity refers to defense that are present at birth. Innate immunity first-line defense includes skin and mucous memories, and the second-line defense includes natural killer cells. Phagocytes, inflammation, fever, and antimicrobial substance. Response of the innate system are activated by protein receptors in the plasma memories of disease cells. Among these activators are toll-like receptors. These TLS attend to various compounds commonly found on pathogens that are called pathogen-associated molecular patterns, PAMPs. Examples include the lipid polysaccharide LPS of the outer membrane of the gram-negative bacteria, the flagellin in the flagella of the metobacteria, the peptidoglycan in the cell wall of the gram-positive bacteria, the DNA of the bacteria, and the DNA and RNA of viruses. TLRs also attend to compounds of the fungi and parasites. Many leukocytes detect repeat chemical patterns found in macromolecules. These unique microbial signatures are referred to as PMPs. The PMPs form specific squeeze-within proteins, polysaccharides, lipids, and nucleic acid, for example, bacteria's flagellin, lipoic acid from gram-positive bacteria, bacterial LPS, peptidoglycan, virus, nucleic acid, dsRNA, or oxalate CpG modifiers. The receptors that bind to PMPs are both soluble and membrane-bound. They are called pattern recognition receptors, PRRS. PRRS that are soluble include acute phase proteins, mannose-binding proteins, and C-reactive protein, CPR. The membrane-bound PRRS are found on the phagocyte cells, enabling them to distinguish between potentially harmful microbes and other host molecules, and importantly, respond by ingesting and degrading the source of the PMP. One PRR type is the monolectin protein, which binds to specific carbohydrates produced by pathogens. One malic-lectin PRR is the mannose receptor, found on dendritic cells and macrophages. Another type of PRR is soluble and remains in the cytoplasm of the phagocytes, known as nucleotide binding and oxidation domain receptors. These sites endogenous methylates and regulate programmed cell death. NLS also detects cytoselect PMPs. TLRs recognize and bind unique PMPs of different classes of the pathogens, and subsequently communicate that binding to the host cell nucleus to initiate protease gene expansion and host response. A number of memory-bound pattern recognition receptors pepper the phagocyte's surface, so as to readily engage their lines when the phagocyte explores its environment. Still, other PRRs remain in cell cytosol the intracellular environment for editance of the evasion or demand. Phagocytes use PRRs to detect PMPs and DMPs on microorganisms and host cells. Memory-bound and cytoplasmic PRRs transduce signals via the same molecular pathway to activate the inflammatory response. TLR4 signals the presence of the bacterial lipopolysaccharide and heat-shock proteins. PMPs bind PRRs, especially toll-like receptor, mannose receptors, and glucan receptors. PRR binding results in signaling that upregulates cytokines gene expressed through a common alpha-kappa signal transduction pathway. Horseshoe structures of TL3 showing attached sugars and inner structures. TL9 signals the declinated CpG modified presence on DNA, and this RNA released by dying bacteria and viruses. When microbes attract our bodies, we defend ourselves by utilizing our various mechanisms of the immunity. Immunity, also called resistance, is the ability to ward off disease caused by microbes of their products and to protect against environmental agents. Lack of immunity is referred to as susceptibility. In general, there are two types of immunity: innate and adaptive. Innate immunity refers to defense that are present at birth. Innate immunity first-line defense includes skin and mucous membranes, and the second-line defense includes natural killer cells, phagocytes, inflammation, fever, and microbial substance. Adaptive immunity is based on the specific response to a specific microbe. Once a microbe has breached the innate immunity defense, it's the third line of the defense. Unlike innate immunity, adaptive immunity is slower to respond, but it does have a memory component that allows the body to more effectively target the same pathogens in the future. Adaptive immunity involves lymphocytes, called T cells and B cells, and will be discussed in detail in the next chapter. Here we concentrate on innate immunity. Thank you for your attention.